previous session, we have seen about the translational significance of animal cell culture part 1, in which we have seen about vaccines, recombinant proteins, monoclonal antibodies and biopesticides. In today's session, we will see about translational significance of animal cell culture part 2, in which we will see about gene therapy, stem cells and 3D cell culture model system. Now, let us see about translational research and its significance for animal cell culture. Cell culture refers laboratory methods to enable the growth of prokaryotic and eukaryotic cells under physiological conditions in vitro. Translational research is a process of applying basic biology knowledge and the knowledge from clinical trials to devices and techniques that address the critical clinical need. Unlike applied sciences, translational research is specifically designed for the improvement of health output. It is indeed uses integrative experts team who focus on transferring important information from the laboratories to the doctor's office and hospitals. It is a bench to bedside research bridge where scientists who study biology and genetics partner with informaticians, engineers, pharmacists, community members, policy makers and others. Now let us see about gene therapy. Gene therapy refers to the insertion, removal or alteration of a therapeutic or a working copy of gene to cure a disease or a defect or to slow down the progress of disease in order to improve the quality of life. The human genome map was the biggest step to study the human diseases and illness in a different way. Gene therapy holds greater promise. However, the task of transferring genetic material to the cells remains enormous technical challenge. Gene therapy requires ex vivo cell cultivation and adaptation from laboratory for a closely clinical relevant state. The development of animal cell culture technology is imperial for the advances of the gene therapy. The monogenetic diseases caused by single gene defects such as cystic fibrosis, muscular dystrophy, hemophilia and sickle cell anemia are the primary targets of the human gene therapy. In gene therapy, the first step is to identify the faulty gene. Then it is followed by the isolation of gene and generation of construct that express correct expression. Integration of gene followed by delivery of genetic material ex vivo or in vivo is very crucial for the success of gene therapy. In general, there are two types of gene therapy, in vivo gene therapy and ex vivo gene therapy. In vivo gene therapy, the genetic material is introduced directly to the host at the target site, whereas in ex vivo gene therapy, the cells are treated outside and they are expanded and introduced back to the host at the target site. Now we see about clinical correlation of gene therapy. Number of clinical studies and trials have been approved and are being conducted worldwide for gene therapy. From 1989 to present, there have been 500 clinical studies reported, of which most of the studies, I would say 70% are intended for cancer treatment. The very first gene therapy product was Genticin. It is produced by Shenzhen Gene Tech of China company. This Genticin is used for head and neck carcinoma. The tumor suppressing gene P53 in a recombinant adenovirus expresses protein P53 that controls and eliminates tumor formation. The production of Genticin uses SBN cell line, which is a subclone of human embryonic kidney cell line 293. Now we see about the viral vector propagation of gene therapy. After the decades of effort, now gene therapy reached its mass limit. Now FDA, that is Food Drug and Administration, have approved two viral based gene therapies, that is adeno associated virus based gene therapies. One is Luxturna. This is used for inherited retinal dystrophy, and another one is Zolgensma, which is used for pediatric inherited spinal muscular dystrophy. The new Imulgic is used for the treatment of cutaneous, subcutaneous and nodal melanoma. Indeed, there are number of gene therapies in FDA approval pipeline, and we expect by 2025 there would be 10 to 20 new gene therapies. This gene therapy relies on viral vectors such as adeno-associated virus or lentivirus, which used for the delivery of designed gene sequence. The cell culture and viral vector products destined for gene therapy should meet high standards of manufacturing quality, safety, persistence, efficiency. Now, let us see about the translational significance of stem cells in animal cell culture. Stem cells are a group of unspecialized cells that can differentiate into any type of cell or tissue and become a specialized cell. The important characteristics of stem cells are their ability to self-renew and differentiate into any type of cells. Indeed, these stem cells can renew themselves to become cells of more precise function. In recent years, stem cell technology has become a major breakthrough in the field of medicine. The property of turning a cell into another specialized cell made the researchers to think about the usage of stem cells to create fully functional tissue or organ to replace damaged or diseased tissue or organs. Now we see about culturing human embryonic stem cells. Human embryonic stem cells are grown on nutrient broth. We use usually mouse embryonic fibroblasts as feeder layers. They give continuous growth of these embryonic stem cells without make them differentiation. This mouse embryonic fibroblast place at the bottom of the culture and provide sticky surface to the attachment of the cells. Moreover, these cells give nutrients into the medium. In recent days, there are many methods developed as animal-free feeder layers in which we use human embryonic fibroblasts and adult fallopian tube epithelial cells as feeder layers along with serum-free medium. More recently, methods have been developed to culture human embryonic stem cells without feeder layer. Matrigel from BD Science has been used to coat the cell culture plate for efficient attachment and differentiation. This matrigel is used for normal and transform anchorage dependent epithelial and normal cells. This matrigel is actually gelatinous protein mixture. This protein mixture is isolated from mouse sarcoma tumor cells. This matrigel resembles the complex extracellular environment present in the most of the tissues. Now let us see the applications of stem cells. The stem cells are advantageous in several ways. These stem cells, particularly adult stem cells, we can attain from bone marrow, adipose tissue, dental tissues and many other tissues. These stem cells are actually a promising therapeutic tool for many incurable diseases. Doctors in the present days use adult hematopoietic stem cells to cure leukemia, sickle cell anemia and other immunodeficiency diseases. Moreover, the stem culture is also useful for the treatment of cardiovascular diseases, type 1 diabetes and brain diseases. Now let us see the translational significance of 3D cell culture model. As you all know, a major milestone in the biological sciences is the establishment of tissue culture technology where live animal cells can be cultured and grown in aseptic environmental conditions. Traditional cell culture, also known as two-dimensional cell culture, is grown in monolayer culture on a flat, rigid surface. Since their development, several advancements have been developed to improve the cell culture media and other biological materials used in the cell culture. These improvements have been shown valuable for cell-based analysis, and this is due to the amalgamation between modern analytical techniques such as fluorescence, electrochemistry and mass spectrometer. This 2D cell culture does not provide adequate in vitro condition where other cells round the cells in 3D environmental condition. Cells under in vivo conditions continuously produce and consume oxygens, nutrients and other molecules. These dynamic distributions are absent in 2D cell culture. Moreover, 2D cell culture does not recapitulate 3D environment, functional and living of functional tissues, and moreover, the interaction between the surrounding tissues are absent. Recent advances in quantitative and system biology and other recent imaging techniques allow the visualization of living cell grown in normal physiological 3D environment. Cells grown in 3D culture highly mimic the dynamic and complex of in vivo conditions. Moreover, this 3D culture provide physiologically relevant and closest biomimetic environment, helps cell differentiation and also improves cell function. 3D cell culture model shows greater promise for applications in various research fields which include cancer cell biology, stem cell research, drug development and various cell-based analysis and devices. Recent developments in the transition of two-dimensional cell culture model to three-dimensional cell culture model provides most promising applications for many industries. However, cost of automation and easy to use readout systems are still the key concerns. 3D cell culture model has gained greater momentum when integrated with microfluid technology. Microfluid technology is characterized by manipulation of fluidics at micron level for the improvement and development of diagnostics and cell culture research. This microfluidics is an important tool because it provides microenvironment for vasculature and thus becoming important tool for cell culture research. Now let us see the applications of microfluidics. This microfluidics technology has emerged as a viable and robust platform for tissue engineering and regenerative medicine. It is a multidisciplinary field which aimed at replacing or repairing damaged or diseased tissue and organ. It also helps to create in vitro models that mimic physiological conditions. Microfluid technology has a wide variety of clinical applications. Successful clinical applications include organ-to-chip technology, which is based on microfluid perfusion device used for regenerative medicine. On-chip tissues include tissues of liver, kidney, lung, intestine, muscle, fat and blood vessels, and also including tumor models. Other important clinical applications of microfluid technology include the chip-based platform, which is used for culture of cells and also cytological or toxicological studies. At the end of this section, we have come to the end of this video lecture series. In this series, we have seen about basic concepts of animal cell culture, types of animal cell culture, cell growth and cell viability, cell culture media, characterization of cell lines, advantages, disadvantages and bioethical issues of animal cell culture, and translational significance of animal cell culture, in which we have seen about vaccines, recombinant proteins, monoclonal antibodies, biopesticides, stem cells, gene therapy and 3D culture models. I hope you have all understood about the basics and applications of animal cell culture. In future, we will see about more interesting facts or topics in biotechnology. Thank you.